we're going to be reviewing genetics over the course of the next unit, and I really love this unit, so I hope you're going to have fun with it. Genetics is just the study of heredity. So that all starts off typically with sexual reproduction and the joining of an egg and a sperm, or two haploid gametes, come together to make one diploid zygote. So fertilization is the joining of two haploid gametes to make one diploid zygote. What does haploid mean? Haploid is a half cell. So you have gametes, which are sex cells. They have half the genetic information. But when two sex cells come together to make a zygote, then it's going to be a diploid cell, or it's going to have two halves, and that makes a whole. So then it'll have a whole set of genetic information. A trait is a specific characteristic that is coded for in your DNA. A trait is a specific characteristic coded for in your DNA. An allele is a variation of that trait. So for instance, if I was looking at the trait of eye color, I might have some different alleles. I could have blue eyes, brown eyes, green eyes, or violet eyes. So an allele is a variation that you can have in a trait. A hybrid is somebody who has more than one allele for a specific trait. So for instance, I have blue eyes. I got blue eyes from my dad. I got blue eyes from my mom. That's why I have blue eyes. My daughter has brown eyes. How'd that happen? Well, she got brown eyes from her dad. But I only have blue eyes, so I could only give her what I have. I can't just make up new traits. I can't say, oh, I want a purple-eyed baby and give her a purple-eyed trait. I can't. I can only give her what I have. So I gave her blue eyes, but her dad obviously gave her brown eyes, or else she wouldn't have brown eyes. So that means that she's a hybrid for the eye color trait. She has two different alleles. One of those alleles was dominant. Which one was dominant? Was it brown or blue? It was brown. It was brown because she's showing the brown colored allele even though she has the blue colored allele, which means one of the alleles was dominant over the other. So which one was recessive? Which one doesn't show? That would be the blue-eyed color gene. So Gregor Mendel is considered to be the founder of modern genetics. But who was this guy? Well, he was a monk. He lived in what is now the Czech Republic, and he was also a teacher, a high school teacher. Fancy that. He was in charge of the monastery gardens, and as part of his teaching curriculum, he and his students would grow pea plants. He used pea plants for a lot of different reasons. First of all, they're edible, and some people think they're delicious. Not me. But they also grow quickly. They had lots of variations in the traits, and he was just looking at the inheritance of different traits in the pea plants. Why would he do that? Well, they knew that if you took a big horse and you bred it with a big horse, you get a big horse. And if you took a fast horse and you bred it with a fast horse, you get a fast horse. So they also were doing that sort of breeding with plants. And he just wanted to figure out, well, why do things get inherited the way they do? Why are traits passed on to offspring? And so that that method, that train of thought is what led him to discover genetics. So Gregor Mendel, he was able to manipulate the way that the plants fertilized each other by controlling fertilization processes. So plants have two different parts. They have the male parts and then they have the female parts. The male parts consist of this anther that's right here, and that's what produces the pollen. Pollen is pretty much plant sperm. And the pollen will then stick onto the sticky stigma, and it'll go into the ovaries of the female portion of the plant. So here's the female portion of the plant, and it'll fertilize the eggs that are inside the ovaries. So what he was able to do is he was able to create this whole line of self-pollinated plants. So the way that plants make sure that they don't self-pollinate and they don't clone each other is the male parts mature at a different time than the female parts. So what he would do is he would just wait for the male parts to mature, he would collect the pollen, and then when the female parts mature, he would fertilize the plant. And he kept doing that and kept doing that until he got what he called true breeding plants. Now, true breeding plants are plants that only have offspring that have that specific trait. It doesn't matter if you breed them with themselves over and over and over again, there's not going to be any variation. So if you took, like, let's say a yellow pea, and he crossed it with a yellow pea, the offspring would always be yellow pea. So he made sure that he had these pure plants, these plants that were pure for a specific trait. He didn't really know why at that time, but he called them true breeding because they always bred true to the trait. The role of fertilization in his experiment was really important. In some plants, he cut out the female organs and he just left the male organs, and then in some plants he just left the female organs and he cut out the male organs. That way he could make sure they didn't accidentally fertilize themselves, and he can control the male to female interaction. So he could cross plants that had one trait with a plant that had another trait and be very specific about it. He wanted to know if you could predict, if I have a plant that has yellow peas and I cross it with a plant that has green peas, what are the offspring going to look like? And he wanted to see if there was a way that he could actually predict the outcomes. So he did a lot of cross-fertilization. And in cross-fertilization, you take one pure breeding plant for a trait, and then you cross it with another pure breeding plant for the same trait, but a different allele. So he would take one true breeding yellow pea, cross it with one true breeding green pea, and then he would take a look at the offspring. The offspring from that cross is called a hybrid because it has to have two different alleles for the same trait. When doing genetic crosses, we call the original pair of plants the P, or parent generation, and then their offspring are called the F1, or the filial 1 generation. Filial means family. So the parent generation, if he took a yellow pea and he crossed it with a green pea, the outcome would be the F1 generation, but he would have true breeding yellow crossed with true breeding green, the parents, creating the offspring, the F1 generation. And this is what happened. If you look, the yellow and the green always had yellow babies, and the round and the wrinkled always had round babies, and the gray and the white always had gray babies, and you can take a look at the chart below to see the different outcomes that he got. So why did the traits disappear in the F1 generation? Well, Gregor Mendel thought that there was a lot of really interesting information that came from this experiment. He drew two conclusions. His first conclusion formed the basis for current understanding of inheritance, and that was an individual's characteristics are determined by factors that are passed from one parent generation to the next. Scientists call factors that are passed from one parent to the offspring a gene. Each of these traits Mendel studied was controlled by one gene that occurred in two contrasting varieties. So remember I said that eye color had 4 alleles? Well, he looked at only traits that came in 2 alleles to make it easier for him to see the outcomes. These gene variations provided different expressions or forms of each trait that were easily observable. So he was really careful to pick traits that only had 2 variations, and the variations were easily observable. The different forms of these genes, if you remember, are called alleles. He found that some of the alleles came in dominant and some of them came in what he called recessive forms. So he established this principle of dominance. This principle states that an organism with at least one dominant allele for a particular form of a trait will exhibit that form of the trait. What do you mean by that? Well, if you've got dad and mom coming together to make an offspring, and dad gives the dominant trait and mom gives the dominant trait, the baby will have the dominant trait. If you have dad and mom come together and dad gives the dominant trait and mom gives a recessive trait, the offspring will still be dominant because the recessive will not show. But if dad gives the recessive and mom gives the recessive, the baby will show the recessive trait. So the only time a recessive trait shows is if they have one allele from each of the parents that's recessive to form the set of alleles, which is going to be a set of recessive traits. So in Mendel's experiments, the allele for tall plants was dominant and the allele for short plants was recessive. How did we know? Well, if you look here at plant height, you can see if a tall plant crossed with a short plant, all of the offspring were tall. That means tall was dominant. So, tell me, what about seed coat? Well, let's look. We've got a gray crossed with white. All of the offspring are gray. That's right. That means that gray is dominant. Let's look at pod shape. I'm going to let you do this one on your own. Smooth crossed with constricted is going to have what offspring? So which one's dominant? Smooth, right, because the offspring are smooth, so that must be the dominant trait. So when Gregor Mendel did this experiment, he not only crossed a pure breeding tall with a pure breeding short and saw that the short trait disappeared, making him figure that one trait was dominant to another, but he also decided that he would cross the F1 generation with other offspring from the F1 generation. So he crossed one of those F1 tall plants with another F1 tall plant, and what he ended up with is all of a sudden the short trait showed back up. So when the short allele showed back up, it was suggested that the allele for shortness was separated from the allele from tallness. This is segregation. Mendel predicted that each trait that the parent carried had an equal chance of being passed on. So pretty much what he's saying is that even if dad had tall and short, that he would make half of his sex cells tall and half of his sex cells short-ish, so it would be about 50-50, but it would just be randomly distributed. So what would you expect in the F1 generation? What would you expect them to be inside of their genes? Would you expect them to carry both short and tall genes? Would you expect them only to carry tall genes or only to carry short genes? Well, Mendel looked at that and he said, well, they must be carrying both genes, or else the recessive wouldn't be able to show up in the F2 generation. And that's when things got interesting. Gregor Mendel came up with these charts. They're called punnett squares, and he used them to try to track and make predictions about what would happen during crosses. So if you look at this chart, on the top is one parent. It's the true breeding tall parent, which means it only has the dominant allele, the big T, and the big T. On this side, you see the little t and the little t of the true breeding other parent. Okay, if you put them into a chart like this, you can see that if the little t comes together with the big T, you're going to get big T, little t offspring. And it's going to happen here, and here, and here, and here, and here, and here. So when he did a punnett square for the second generation, he figured out exactly how the short trait showed back up. If you have a parent that has big T and little t, like the offspring did before, and a big T and a little t, and they crossed, and each gamete could only pick big T or little t in about 50-50%, big T, little t for the eggs, 50-50%, and the big T comes together with the big T, and you get big T, definitely tall, big T, little t, tall, big T, little t, tall, because tall is dominant, and then you get little t, little t. That's where we got our short plant. Okay? We call this homozygous dominant. So it means that the zygote, homo means the same, so the zygote has the same dominant characteristic from each parent. It means it's true breeding, dominant. Over here, you can see this one is homozygous recessive. It got a recessive trait from mom, a recessive trait from dad, and so it only carries the recessive trait. So homo means the same in the zygote, and it's recessive. The other option is to be heterozygous. So you might look like you're tall, but you still are a carrier for a different trait. And in this case, we would call them hybrids. Okay, so we've got heterozygous here and heterozygous here, but they still look tall. So if I was just to look at what they looked like, what I can see with my bare eyes, I would see that I have 1, 2, 3 that are tall, 1, 2, 3, and 1 that is short. So guess what? Gregor Mendel got about 75% that were tall and 25% that were short. So every organism has a genetic makeup that plays behind the scenes of your observable characteristics. So you've got some genes that are behind your eye color and your attached or unattached earlobes and your hair color and how wavy it is. It's all coded for by genes, and those are behind the scenes. So the things that you see that we say are expressed or are on the outside, that's the phenotype. I kind of remember phenotype as pheno is face. So pheno is what you're going to see at the face value. It's what's expressed. It's what's on the outside. Genotype, on the other hand, is what's going on behind the scenes. They have brown eyes. Do they have big B, or do they have big B, little b? It's hard to tell. So in order to describe genotypes, once again, we have homozygous dominant, 2 big B's, heterozygous, big B, little b, or homozygous recessive, little b, little b. So, how many different genotypes were in that F2 generation? Well, there was 3. All of them were represented. How about in the F1 generation? There's only 1. All the F1 generation was heterozygous. Okay, I want you to get out your Bikini Bottom genetics worksheet. Pause until you are all ready with your Bikini Bottoms packet. Okay, so I want you to look at question number 1 in the front of the packet. It said for each genotype below, indicate whether it's heterozygous, he, or homozygous, ho. Ready? So big T, that would be homozygous. They're the same. Big B, little b, heterozygous, he. So now let's keep going through, and we're just going to go across the rest. Okay? So big D is ho, then he, then ho, then ho. On the next line, starting off with big D, little d, that's he, ho, ho. Good. All right. So which of the genotypes in number 1 would be considered purebred? Would it be the homozygous or the heterozygous? Homozygous. I hope you got it right. Which of the genotypes in number 1 would be hybrids? Heterozygous. Good. The next question says, determine the phenotype for each genotype using the information provided about SpongeBob. Yellow body color is a dominant to blue. So, big Y, would that be yellow or blue? Yellow. Big Y, little y, yellow. little y, blue. Good. Now I want you to do the next one on your own. We'll check it in just a minute. Big S, that would be square. Big S, square, little s, definitely a round pants. All right, so for each phenotype, give the genotypes that are possible for Patrick. Big T is tall head, and it's dominant to short, which is little t. So if you were tall, you could have only these two possible genotypes. Good. Homozygous dominant, big T, or heterozygous, big T, little t. Excellent. What about if you're short? What genotype or genotypes could you have? You're right. You could only have the little t. Excellent. Do the next one below that. Pink body color, big P, is dominant to yellow, which is little p. So pink body would be big P, or big P, little p. Yellow body would be little p. Good. I think you're ready to do Punnett squares. So, to do a Punnett square, the first thing that you need to do is you draw a box. And then you've got to figure out, well, what are the gametes for the parents? So let's say parent 1 is yellow. And you know that yellow is dominant to green. But you don't know if parent 1 is homozygous dominant or heterozygous. Well, guess what? It's either going to tell you in the question, or it's going to give you a clue. It'll say one of its parents was green, or something like that. Sometimes these questions can be super fun because they're like a mystery or a puzzle, and you've got to like go back in the family history and figure out, you know, what the offspring looked like. It's kind of interesting and fun. But in this example, both parents are heterozygous for the yellow P allele. So they just told us straight off, both parents are heterozygous, which means both parents are big Y, little y. But each gamete can only have 1/2 of that. So, in this case, each gamete is put in above each one of the boxes that the little box set that you made. So, you make the little box set, you just make a square, and then you put an X in the square, and then above it on one side, you're going to put the big Y, and on one side, you're going to have the little y. So, if this is the dad and this is sperm, then half the sperm would have big Y and half the sperm would have little y, and then they would just be all mixed up. If this was the mom and she's making eggs, half the eggs would have big Y and half the eggs would have little y, and then they would just be randomly dropped whenever she's ready. Okay? So, if this egg came together with this sperm, we would get big Y. If this came together with this, then we would get heterozygous, big Y, heterozygous, little y, homozygous recessive. So if I was to look at the phenotypes, I would say that I have a 1, 2, 3 to 1 phenotype of yellow to green. Excellent. You got this. So I've got a green pea plant, big G, little g, and it's crossed with a yellow pea plant, little g. So, what's the outcome going to be? I want you to go ahead and do these on your own. Write them down on like a side of piece of paper, a little scratch or something. And I want you to fill them out, and then we'll review in just a moment, okay? So push pause while you're waiting. Excellent. So, for number 1, you should have big G and little g coming together, and you should have little g and little g on one side. Now I know I'm going to have somebody go, but could I put little g up here And big G, little g over here, yes, it doesn't matter. It doesn't matter if you make this the sperm and this the eggs, or if you put the sperm over here. It doesn't matter. As long as you have the one heterozygous parent and one homozygous recessive parent. Okay? Your genotypic outcome would be 2 heterozygous and 2 homozygous recessive. And your phenotype would be 2 green pea plants and 2 yellow pea plants. On the next one, you have homozygous tall crossed with a heterozygous. So the outcome is going to be all tall, right? The phenotype would be all tall, 100% tall. But you're going to have 2 homozygous dominant and 2 heterozygous. So 2 heterozygous, 2 homozygous dominant. On the final one, you have little t crossed with little t. So you have 2 recessive parents crossed together. So you've got little t and little t. All of the offspring are going to be homozygous recessive. So all of the offspring are going to be short. I would like you to please finish pages 1 through 4 in your SpongeBob packet. We're just going to do pages 1 through 4. Okay, so pages 1 through 4 in your SpongeBob packet, and then you're done for the day. Okay, we are going to review this. You're going to correct it. You will not be graded on whether it's right or wrong, but I want you to practice and I want you to do your best, okay, so that we can find out where you're at and what we need to practice more. Okay, so finish pages 1 through 4 in your SpongeBob SquarePants packet, or sorry, Bikini Bottoms packet, and then you're done for the day, and I hope you have a great one. Take care.